Hi, it's Sophia here, and today we're going to talk about the cell cycle. Hopefully, by the end of this video, you'll have a greater understanding of what the cell cycle really is, what checkpoints are, what they do, and what are some stop and go signs that are given at each checkpoint, and proteins and enzymes involved in these processes. And finally, what happens when the cell cycle stops working. So let's get started. So what is it? What is the cell cycle? The cell cycle is an illustration of how a cell spends most of its life, resulting in division that produces two daughter cells, which are identical to the parent cell. It is obviously cyclic in nature, and as you can see, most of a cell's life is spent during interphase. Interphase is the daily life of a cell. In this phase, the cell grows and does cellular work, and it prepares for mitosis in the M phase. Interphase is divided into three stages, G1, S, and G2, and there is technically a fourth stage called G0, but we'll talk about that later. In G1, or growth 1, cellular contents, except for chromosomes, are duplicated. In S phase, DNA is synthesized and replicated, so all of your chromosomes are going to be duplicated by the cell. In G2 phase, the cell will double-check the duplicated chromosomes for error and will make any needed repairs. And this is the final phase of interphase as the cell moves to cellular division. And as you can see, these are all going to be components of interphase. So M phase is mitosis, and mitosis is the process of nuclear division divided into four stages: prophase, metaphase, anaphase, and telophase. And prophase separates genetic material, and the nuclear membrane dissolves, and chromatin is condensed. In metaphase, chromosomes are at their most condensed state, and chromosomes line up along the metaphase plate. In anaphase, sister chromatids are liberated and they move to opposite sides of the pole, and in telophase, sister chromatids are going to reach the opposite sides of the pole and the nuclear envelope starts to form. So here you can kind of see anaphase and telophase happening, and right here you're going to be able to see cytokinesis right here. And cytokinesis is going to be the actual cellular division along with the cellular contents in the cell, obviously. And that's going to be part of cytokinesis. And there is also an additional phase in interphase that is sometimes overlooked, and it's viewed as an extended G1 phase. It's called G0, and G0 is a stage where the cell is under cell cycle arrest, as people like to call it, and it is neither dividing nor preparing to divide. It's just not doing anything, basically. So checkpoints. Checkpoints in the cell cycle serve as control mechanism which ensure proper cell division. Each checkpoint acts as a halting point during which all conditions are assessed in the cell. The G1 checkpoint is the point at which the cell is committed to enter the cell cycle. In other words, if this checkpoint is passed, there is no going back. If it does not pass this checkpoint, the G1 phase is just going to be extended, or it will go into cell cycle arrest. The G2 checkpoint is going to follow DNA synthesis in S phase, so correctly it'll check for DNA damage. This checkpoint ensures that the cell underwent all necessary changes during S phase and it is ready to divide. And the G2 checkpoint serves a very important role, and it is to prevent cells from entering mitosis with genomic DNA damage. And finally, the M checkpoint. The M checkpoint occurs at the point in metaphase where all the chromosomes should have aligned at the metaphase plate under bipolar tension, and when this tension is sensed, it initiates anaphase entry. Cyclins and CDKs. Cyclins or CDKs are crucial to the cell cycle because through phosphorylation, CDKs signal the cell that is ready to pass into the next stage of the cell cycle. And as their name suggests, cyclin-dependent kinases are dependent on cyclins, which are another class of regulatory proteins. Cyclins bind to CDKs, which activate the CDKs to phosphorylate other molecules. So in this graph, you will be able to see how different cyclins change their concentration along the cell cycle, and how these different cyclins will be able to react differently in the cell cycle, which makes them able to control progression of cells along the cell cycle. So stop and go signs. And as studied previously, cell division is controlled by complexes made of several specific proteins. A specific complex is mitosis promoting factor, or MPF. MPF promotes mitosis by phosphorylating various proteins. And as you can see here, as MPF is degraded, the M phase stops, so mitosis stops as it degrades. And the CDK component of it is then recycled to be used in G1. And this cyclin accumulates throughout S and G2 phases, and these accumulated molecules, they combined with the recycled CDK and produced enough MPF to pass the G2 checkpoint, as you can see here. So an example of an external factor that promotes cellular division are platelet-derived growth factors, or PDGFs, which are proteins released by platelets that stimulate cell growth. This experiment on the action of PDGF is set up as follows. A sample of human connective tissue is cut up here, and then enzymes are used to digest the extracellular matrix in the tissue, and then these cells are then transferred to different culture vessels. In this culture vessel, it does not have PDGF, whereas in this culture vessel, it does have PDGF. And the results indicated that without PDGF, cells fail to divide, whereas with PDGF, cells do proliferate. So what happens when these control mechanisms stop working? Well, as you can see, cancer is going to be one of the effects of the cell cycle, damaging, basically. And some control mechanisms are contact stimulated, such as density-dependent inhibition, where crowded cells stop dividing, and anchorage dependence, where the phenomenon in which cells must be attached to a substrate in order to divide. And here you can see extreme cell proliferation, which will cause a tumor. And if a cell or group of cells does not have these properties, such as anchorage dependence or density-dependent inhibition, it'll cause uncontrolled cell proliferation, shown here, which leads to cancer. Cancerous cells may not need growth factors to grow and divide, so they're basically growing on their own. If and when these cells stop dividing, they will not do so at design checkpoints. They will do so basically at random points whenever they want. But the problem with this is that the enormous cell cycle control system creates malignant tumors that can then metastasize, as shown right here, where it means that the cells are exported, the cancer cells are exported to other parts of the body. Okay, so by now you should hopefully know a lot about the cell cycle, its control system, and what makes it so special and so important in eukaryotic cells, in eukaryotic organisms, and what happens when something goes wrong with all of the components that control the cell cycle. So I hope that was helpful.